Good evening. Thank you so much for joining us tonight. My name is Cherrie Demayo, and I am the nurse coordinator for the UCSF Neuro-Oncology Gordon Murray Caregiver program. And tonight I will be hosting this event, and I'll be your hostess. So this is our 6th annual already Adam Clar Nursing Lectureship.
That's what I said. I'm like, oh.
Yeah. And this is our agenda for tonight. And then we'll have time afterwards for Q&A and for everyone to be able to ask their questions. So, as you may have read on your programs on the table there, and if you've joined us in previous lectures, you know Adam. Adam Clar is the namesake of this lecture. He's a nurse, and he was diagnosed with a brain tumor in 2004. Adam was treated here at the UCSF Brain Tumor Center, and while undergoing care and treatment with our team, he was also simultaneously pursuing a career change to become a nurse. He was exceptional in his pursuit in practicing nursing, and in 2014, he successfully graduated with honors from Samuel Merritt University School of Nursing. That's about 10 years from when he was initially diagnosed. Adam was an excellent nurse and very much loved by his colleagues and the patients that he served. Sadly, just as he was about to start his dream nursing job, Adam's tumor returned, and he sadly passed away at his home in Santa Cruz 6 months later at the age of 39. So as both a nurse and a patient, Adam understood the importance of continuing education for nurses and all healthcare professionals. So the Adam Clar Neuro-Oncology Nursing Lecture Fund was Adam's idea and wish. In 2019, we held our inaugural lecture, and we have been able to fund and offer this lecture to UCSF nurses and staff for the last 6 years now. So tonight, we honor Adam and extend our gratitude to Adam's family who have joined us here tonight, and for really fulfilling Adam's wish and for sharing his love of nursing and continuing education with all of us. So thank you. So in addition to this lecture, the Adam Clar Nursing Lecture Fund also helps support our nursing staff's attendance to scientific and educational meetings and conferences. This past year, 5 of our team members were able to attend the Society for Neuro-Oncology annual meeting that was held in Hawaii. This fund helps our staff gain knowledge about scientific and medical advances in the field of neuro-oncology, which we then bring back to our clinic. These are some of the testimonials from our nursing staff. So again, thank you to everyone for your continued support. This means a lot for our staff, and it means a lot for the nurses who are here as well. And so as we remember Adam and his legacy, I would also like to honor everyone here. All of you work with our neuro-oncology patients and families. And this work does not come without heartache, but you continue to show up every day. So we're also here to honor all of you. Along those lines, I would also like to take this moment to present the Adam Clar Neuro-Oncology Nursing Award. And this award is presented to a nurse who exemplifies outstanding leadership, dedication, and service to the neuro-oncology patient population and the neuro-oncology field. So our first awardee, Sharon Lamb, who is here with us tonight also, thank you for being here, is one of the nurse pioneers in the neuroscience field and also one of Adam's nurses. She graciously donated her bronze ribbon sculpture, which you'll see here and on the table here. And so every year since 2022, each awardee passes the sculpture along to the next awardee, and they get to keep it for a year. So some of our awardees are shown here. Martina is here as well. So thank you for being here. And this year, we are honoring nurse leader Dr. Mary Lovely, who has dedicated her nursing career to the care of neuro-oncology patients and their families. She earned her bachelor's and master's degrees of science in nursing from the University of Washington. She began her nursing career at San Francisco General Hospital in the Department of Neurosurgery. Through her work, she became intimately involved in research and helping patients diagnosed with a traumatic brain injury. This led her to become a neuroclinical nurse specialist at the Veterans Hospital, where she began working with patients diagnosed with a brain tumor. In 1996, Dr. Lovely earned a PhD here at the UCSF School of Nursing, focusing on symptom management and quality of life for brain tumor patients. Dr. Lovely's work since then has spanned the nonprofit and industry settings where she provided guidance and expertise on how to best support brain tumor patients. Dr. Lovely is well known nationally and internationally and has given lectures to patients, families, and healthcare professionals about brain tumor care. She led a research study with a network of neuro-oncology nurses throughout the United States. Since her retirement, Dr. Lovely continues to be a brain tumor patient advocate and co-facilitates our UCSF Patient Brain Tumor Support Group. She has facilitated this group for the last 28 years and continues to do so we're very grateful. And what I've shared does not begin to capture the breadth of Dr. Lovely's contributions to the field of neuro-oncology and the impact she's had on the lives of countless patients and families. Her dedication and passion for improving the lives of neuro-oncology patients and their families are qualities I deeply admire. Please join me in congratulating and welcoming our 2026 Adam Clar Nursing Awardee, Dr. Mary Lovely.
Thank you, Cherrie. It's just really, it's nice. It's nice to be here, and thank you so much for giving me this award. I was shocked when you told me. Oh. and thank you to the Clars for remembering the importance of nursing and what nursing in neuro-oncology is all about. It's really means a lot to me. I just want to say a couple other things. Actually, I started on a regular neuro floor, and I at University at Harborview in Seattle, and I didn't know much about neuro, but I knew that if I could, if someone said to me, if you can do neuro, you can do anything. So, well, okay. So I and then went to an epilepsy center, and that was very interesting. And then realized I needed more education and got a master's in neuronursing. But when I came down to San Francisco and I was a clinical specialist at the VA, and they said, here, you get to take care of the brain tumor patients. I went, oh my gosh, what an interesting thing. So I got to meet people who had brain tumors over and over again. I even presented him to brain tumor board, and it was an experience. And then I realized there's really not anything for these people. We don't have what happens after they're diagnosed. So I focused on quality of life and symptom management, and it's been a wonderful, wonderful career in really different ways. And all of you many of you have been part of my journey, and I continue to do it as the support group leader, because I find that they're so special, and I'm not ready to say no, I'm done. And I go to those meetings once a month, and darned, if there's someone that's been there 23 years, that really is what keeps me going. And I also am there when somebody's new and they're going, oh my gosh. Or we're there when somebody says, this is going to be our last meeting, but we're there, and they know we're there. I want to thank my partners in crime, what sometimes I feel a lot of times that we're part of the community. We're part of a community branch of UCSF. And so I have today, we have Joni Taylor and Sharon Lamb, who are also, they're retired nurses, and they work in the community with me still, leading support group as well. So I really appreciate that. So that's what I wanted to say there. The other thing is, what really helped me as a neuroscience nurse is to develop a network with other nurses. And that made me feel stronger, because this is hard work, but it's really, really, really rewarding. So, in Margareta. So I really appreciate it, and I'm so glad to be on it today. And Suzanne, you've been part of my brain tumor journey. We've been part of our brain tumor journey nationally, internationally. Been to Australia together. We've been to Italy together, and all these places. But there's people who care about brain tumor patients and families, and we're there to either teach, to work with other healthcare professionals, and it's just wonderful. So we'll just keep it going. Thanks.
So we'll get started with our lecture for tonight, and I'd like to introduce our speaker. We will be hearing from Dr. Melike Pekmezci, a board-certified neuropathologist with a clinical and translational research interest in the molecular classification and grading schemes of diffuse gliomas. Dr. Pekmezci has been a member of numerous professional organizations. She has authored 150 peer-reviewed papers and has authored multiple book chapters, including those in the World Health Organization Classifications of Central Nervous System. She is a member of our team here at the UCSF Brain Tumor Center. Tonight, she will walk us through the process of molecular profiling of adult gliomas and how this may help providers and staff understand and predict how quickly a particular tumor may spread or evolve. As we enter the world of precision medicine, her work is particularly valuable in helping us identify diagnostic and prognostic markers, which then help teams improve management and treatment of symptoms related to a patient's brain tumor. So please welcome Dr. Melike Pekmezci.
Thank you very much. Thank you for the invitation. Thank you for having me here. And thank you for supporting such a lectureship and then award for honoring nurses and honoring Adam. So today's talk is going to be a little different because we are going to basically take a step back and then talk about what happens to the tissues once they are out. And then when we actually work with these tissues, we sometimes think about it as a tissue, but we are well aware that there is actually a person behind those tissues. And rest assured, we actually do pay attention to the patients, not only the demographics like age, where the tumor is coming from, but to the patient. And now whatever we diagnose provide, we know it will affect something. So I'll try to highlight some of those nuances maybe, and then try to not cover everything that is listed on the current WHO book, because that will not be very fruitful. But I will not even cover all the gliomas or glioneuronal tumors, but I will start with this slide because I want to highlight how the evolution, like the glioma classification evolved over time. So first of all, this is a short list of the tumors, mostly gliomas and glioneuronal tumors. And with the new classification, there are a couple that I highlighted there with that red asterisk that actually have a specific mutation after their name. So you have to show that molecular alteration to give that diagnosis now. So it's not just we look at the slide and come up with a diagnosis. And there are a couple others there. Even though the specific mutation, gene alteration is not listed on the name, we know they exist. Sometimes it's not listed because there are more than one of them. But we have to really show those alterations to come up with these diagnoses. And abundant of these tumors, however, have, again, specific molecular alterations. If you tell me a random molecular, not random, but one of those, I can actually pretty much tell you what the tumor is going to look like. There's such a big, strong correlation with that nowadays. In addition, there are multiple newly described entities, because now we do so much more molecular testing on all these tumors. Sometimes tumors that we had hard time classifying in the past, now we know their molecular alterations, and there are new tumors getting added to this list of already long list because of that. And finally, it's not only about diagnosis, but we also know, actually, before getting that, these last two that just showed up in that last group, glioblastoma being the most common adult high-grade glioma, is defined by what does it not have, rather than the genetic alterations it has, because it still is the most common tumor. And it is, I'm not going to say wastebasket per se, but anything that looks high-grade that doesn't have a specific designation or home elsewhere still goes to that group. And that group is the most quickly changing group nowadays, because more and more there are more tests done, and then more subgroups are separated out from that group. And then we can be a bit more specific about what that tumor is actually like, rather than, yes, it is a high-grade glioma that doesn't have IDH. But nevertheless, that's one of the largest groups, and that's the group that is evolving fastest, I would say. And finally, these molecular alterations not only define a tumor now, but it can also give a prognosis. Therefore, if certain alterations are present, we now can say a tumor is a high grade or low grade, which may change the management, just purely based on the molecular alterations they have. So this is a study we've done here way back then, actually before 2021 classification. But this is, if you look at the adult type diffuse gliomas, there is a significant variation in their prognosis. This is an overall survival curve. So over time, what's the percent of patients still alive with that diagnosis? And if we start with an oligodendroglioma IDH mutant 1p19q co-deleted, they have the best overall survival, followed by IDH mutant astrocytomas. And this very bad actor is the IDH wild-type glioblastomas. So this group is basically a mixture of either IDH mutant but morphologically really bad tumors, so this group 3. And there is this other subgroup that looks, it's pretty bad outcomes, even though they do not look histologically high-grade. They behave very much like a high-grade tumor, and that's why current classification basically lets us assign a higher grade based on the molecular alterations, even though histologically they don't look bad. So why do we care? So on that image, you already saw IDH mutation is such an important prognostic factor. So tumors are classified whether they have IDH or not. And there's another layer now that IDH mutation, specific IDH protein inhibitors are available for treatment. This is going to be even more important. So I'm not going to really talk about treatment here while Dr. Chang is here, but in one of these relatively recent clinical trials, they found that patients who received an IDH mutant inhibitor directly has a really better overall survival and event-free, basically event meaning there's a recurrence or something else that they have to do for that patient, event-free survival, as opposed to the patients who receive placebo. So these are working really well. And with that notion, IDH mutation detection is even more important nowadays. So how do we approach diffuse glioma? So now this is even the shorter list. I listed there just for people in case you're interested, what common mutations are present in a given tumor. So in our practice, we look under the microscope to see what the tumor actually look like, and I will show you some images, but also we evaluate whether they have these particular mutations or not. And based on the combination of mutations they have, we can assign which tumor is what. And I will show you examples of the tumors with the highlighted with that red, because they are actually relatively straightforward to diagnose. They have a specific stain we could do, and then diagnosis is often not a mystery. So we start with histology. And the first thing is, well, to confirm it's a glioma. I'm completely skipping that part, but it doesn't always have to be glioma. Once we know it's a glioma, then we do our immunohistochemistry, which is a method that is recognizing specific proteins that are present in that tissue that is probably not present in normal brain. And when we do that, we can make it whatever color we want, honestly, but we love brown. So if I show you some blue and brown pictures, if there's brown, that protein is present in that tissue. And if the protein is specific to that particular mutation, that's it, we confirm there is that mutation. So that's as simple and usually works, but it's not as easy for some other stains maybe. And if that's a question that cannot be answered that quickly, we have to do some gene sequencing to look for specific mutations. And we can also do some additional genetic tests looking for the chromosomal copy number changes. So tumor tissues have DNA, and their DNAs are not going to be normal. And based on the changes in that copy number, we can actually either diagnose a tumor, like the oligodendroglioma, or predict whether they will behave aggressively or not. And we can do that with various tests, and which test to use kind of changes depending on what we are searching for and how much tissue we have. But it is routine practice now. It's not really fancy science. It's available almost everywhere. And then finally, we can also do genetic tests looking for gene fusions. So this is a different type of molecular alteration. And this is not very common for adult type gliomas, but this is one of the main drivers for pediatric type gliomas, like in infants, for example. And finally, nowadays, maybe the biggest hype of the day is the DNA methylation analysis. And this way, it's not looking for a specific gene, but it's looking at the DNA of the entire tumor and trying to match this one tumor to the existing library of tumors and then tell us which tumor that is closest to. So that's another method nowadays. I will try to do this as a case-based discussion, and this is just a couple examples of maybe classic examples of certain tumor types. The first case is a 34-year-old man who presented with visual field cuts, and the imaging, basically there is a big, we call this flare, hyper-intense, big bright white region in the left temporal lobe. And it does not have contrast. So with this imaging, we were expecting that it would probably be a low-grade diffuse glioma based on that morphology. So this is what normal cortex and white matter in the brain look like. And here next to it is the sample from this tumor. So first thing to note, hopefully everyone can appreciate, it's too many cells there, right? We shouldn't have that many cells. And looking at the cells, they have more irregular nuclei. All the purple nuclei they have irregular edges rather than being perfectly round, as you can see on the white matter. But they are single cells infiltrating, we call that, to the existing brain parenchyma. That's what the diffuse glioma means. They are diffusely infiltrating rather than kind of have a pushing border. These are the fancy stains that we do on every diffuse glioma in adults. And like I said, these are immunohistochemistry. So in that very first stain on the upper left, we are looking for the presence of IDH1 mutant protein. And because the protein is there in that tumor, it's brown. On the second picture on the ATRX, when there is an ATRX mutation, it basically deletes the protein. And then instead of having brown, tumor cells have blue. Blue is our counter stain, just to make sure we can see such stuff. So there's ATRX loss and IDH mutation, and P53 is increased when there is a mutation. So these three basically tells me this tumor has 1, 2, 3 mutations, IDH, ATRX, TP53. So it makes this an IDH mutant astrocytoma. And it makes sense. Imaging-wise, we were expecting a low-grade tumor. Histology is low-grade. The age is very common for this type of tumors. Everything adds up. And our stains add up. So this diagnosis is straightforward. I didn't even do a molecular test, right? I only did surrogate markers for molecular. This is one example. It's not always as straightforward though. So there's another example of a 37-year-old, this time the very similar mass on the imaging. Here you can see a really bright T2 bright tumor. This one is taking some contrast, which basically tells us the tumor has some leaky vessels. So when you give the contrast material, it's leaking into the tumor itself, which is usually worrisome a bit more for a higher grade tumor. Histology looks in so many ways very similar. It's again too many cells, and it is infiltrating into the brain parenchyma. This is the normal brain cellularity, just for comparison. And we start finding mitoses. Basically, we can see the cells actually proliferating in front of our eyes. This is more common in higher grade tumors, and we quantify how many of those mitoses we see in a given field. We did the same stains. So this one does not have that particular IDH protein, right? Because it's all blue on the upper left. ATRX is still lost, and P53 is still increased. So just looking at it, I know this tumor has ATRX and P53 mutation, but IDH I couldn't pick up. Does that mean it is IDH wild type? No. So this is where molecular becomes very important, because everything else tells me this tumor should be IDH mutant, but I'm not picking it up with my stain because it is picking up this one particular mutation, but there could be others. So we have to really send this for molecular testing, and it could be done with a single gene sequencing, or instead of looking at these genes one by one, we could do a panel. And at UCSF, we have UCSF 500, which actually has 529 genes in that one panel. So we don't have to do it one by one. But if we were to do one by one, maybe 20 years ago, this was the best available test, we would do Sanger sequencing and find that mutation. But right now, we don't really do that. And just to highlight that further benefit, if we did that, we would call this tumor grade 3 because histologically it has mitoses, and we found the IDH mutation. But nowadays, the classification, like I said, it can escalate increase the tumor grade based on the molecular findings. So we did UCSF 500. It is a 500 something gene, so we don't actually see individual genes. We get like an Excel output, if you will. But you may have seen similar reports. We basically list every single mutation that we think is pathogenic, doing something in that tumor. And this tumor has ATRX, IDH, P53, as we expected, but it also has this deletion of this gene. And I hope it's here, this gene is located right there, and it's deleted. It's basically going all the way down. So all these dots correspond to a single probe and a gene in that whole chromosomes 1 through 23, and like X and Y, 22 and X and Y. And we find this deletion here. So that gene that's deleted is one of the genes that keeps the cells in check, if you will. If the tumor can get rid of that, it can proliferate endlessly. So that's what it's doing. And because that gene is deleted, we can assign this tumor a grade 4 now, because we know it's going to behave more aggressively than a grade 3 tumor. So this patient can be treated properly, right? And this is another example of like why that gene made its way as a grading criteria. This is one of the studies showing that if you just look at the histology for an IDH mutant astrocytoma, grade 2, 3, 4, just looking at on the slide, versus if we can show that gene is deleted, that gene name is CDKN2A. So anytime that gene is deleted, the survival goes down because those patients tumors will grow too rapidly for the patients to kind of wait and see. So they will need more urgent treatment. I just showed this a couple of slides ago because this is the IDH mutant inhibitor treatment, and this study specifically focused on the grade 2 tumors. But I want to point out something here. There is actually very little difference between grade 2 and grade 3 tumors. And the current studies are maybe focusing on, should we really limit the treatment to grade 2, or should we let other patients maybe potentially use this treatment? And that's an ongoing research because we know that they will probably respond, but if you keep adding new mutations into a tumor, will they respond as well? That's an open question. So that's another area I think neuro-oncology is going to focus on in the next few years. Let's move on to a different tumor type. Another 35-year-old presented with seizure. This patient has a tumor essentially located in the parietal lobe here. Again, pretty bright on T2, like all the other tumors. It's again supracellular, too many cells. But this one, I don't know if you could appreciate, the cells have a perinuclear clearing. We call them a fried egg appearance. They look like a bunch of fried eggs on the plate. So this tumor and the nuclei are, as opposed to the other ones I showed, pretty consistently round. Just by looking at it, I am suspecting this will be a different tumor type. It will be an oligodendroglioma. And they have different mutations, but they have to have IDH mutation, and that stain is again brown. So we have IDH mutation, but they do not have ATRX mutation because all the cells retained their ATRX, so that mutation is not there. And P53 is not increased. And I have been kind of skipping the Ki. It's basically a marker that shows us how much the tumor is proliferating. So this one is low, but in a couple cases later, we will see a couple cases with even more proliferation than this. So this tells me this tumor has an IDH mutation, but it does not have ATRX and TP53 mutations. And I was already suspicious of oligodendroglioma, but to get to that diagnosis, we have to do more tests, which is you can do by fluorescence like this. And I'm looking for specific chromosomes in these tumor cells, and we can see the part of the chromosome is deleted. So we have 2 centromeres, we call them, in each cell that is red here, but they only have one short arm of the chromosome 1, not the other. So the second arm is consistently deleted, and this is the chromosome 19q doing the same thing. So these tumors have, we call that 1p19q co-deletion. They are deleted together, and that's what makes it an oligodendroglioma, IDH mutant 1p19q co-deleted. Otherwise, if I couldn't show this, we wouldn't be able to call that an oligo. Why is that important? Oligodendrogliomas have significantly better prognosis than even IDH mutant astrocytomas, and the treatments will be different. Potentially, maybe held, like if the tumor is resected completely, maybe there won't be any additional treatment until the tumor recurs or something unexpected happens. So we don't really do FISH so much anymore because we do next generation sequencing on all of these cases. And this case will show that IDH mutation. Often they have additional things that are expected, so I'm not surprised about that. And this is what 1p and 19q, so short arm of the chromosome 1, long arm of chromosome 19 are deleted. So all the other chromosomes have normal numbers here. These are deleted, right? Another case. So not everything is all about IDH. And you learned a lot about the brain tumors because especially the ones that are in the midline locations, different than the cortex, behave very differently. Part of it is you cannot really take out some of these midline structures. So it has to be a biopsy and maybe potentially more chemo versus more superficially located tumors can be taken out, at least to some aspect. But also physiologically they are different. They have different mutations. So this one is from a 72-year-old patient who presented with altered mental status, and imaging showed a tumor in the basal ganglia, deeper, like in the deep nuclei, deeply located tumor. Hypercellular, but not super hypercellular like the other one. It could have been more cellular than this. Cells are still very atypical, really dark, really irregular nuclei. This is what we see in our first pass on tissue. And if we do our stains like we would otherwise, all of these stains will tell me there is no mutation in IDH, no mutation in ATRX, no mutation in P53. Now, this is not the common type, right? What it does instead is a mutation in that histone, H3, this specific K27M mutation, because it's the same idea. We have a protein that is mutated and we have a stain that recognizes it specifically. So because the tumor is in the midline location, it's a diffuse glioma and it has this mutation, we call it a diffuse midline glioma, H3K27M altered. We are not trying to be poetic, very descriptive, right? So basically whoever reads it will get it. By default, these are considered grade 4. We don't really look for any other histologic features. These tumors need to be treated aggressively. Otherwise, these patients, the tumor will recur basically, partly because you cannot take it out. Let's go to a different example. 27-year-old woman has a tumor. I'm having a hard time with the arrow. But hopefully you can now find the tumor. There you go. This tumor is located sort of deep, but actually still in the hemispheres, just trying to extend from right side to the left side. And it has, basically this imaging says this is a very hypercellular tumor. This DWI shows supercellular. Radiologists probably tell us this is a high-grade glioma based on these imaging features. And it is supercellular, and the nuclei are even darker than everything else I showed you so far. In a different area, it will be even darker, right? So there's a spectrum of how bad things could be, I guess. This tumor, when we do our stains, is also going to be negative for IDH mutant protein, but it's lost its ATRX, so there's ATRX mutation, and it has a lot of P53, so there's probably P53 mutation. But the money slide is this other mutation, same gene group, H3, but a different mutation in a different locus, and we can recognize that with our stain, which makes this a diffuse hemispheric glioma H3G34 mutant. Grade 4, right? No surprise there. This is actually, if you go back all the way to the very first table, this is considered a pediatric type glioma, but this patient is not a kid, but this is more common in young adults or maybe adolescents, so it could be seen in kids. So these are the reasons. So if the tumor basically reads the book and the stains function well, we can get to the diagnosis really in a 2-3 day cycle. But if we need to do molecular tests, if these stains don't add up, we will have to really wait 2-3 weeks maybe. And then if they don't add up, we have to do more, and then it adds. And just to conclude or get close to conclude with 2 older patients, they tend to have different tumors. They tend to have tumors that doesn't have IDH or H3 mutations. So this one is again a bright tumor in T2. And then this one, when we give the contrast, it is showing this ring enhancement pattern, suggestive of leaky vessels, which says high grade, just looking at the imaging. Tumor is really high grade, it's high cellularity, and these nice picturesque areas in the middle that looks like maybe some animal shapes are lakes of necrosis. So the tumor cells are proliferating so rapidly, they're also dying very rapidly, and the other tumor cells are sort of surrounding them. We call that palisading necrosis. It's very like fence-like, trying to limit the necrosis. And tumor is trying to also bring up its own vessels, and we have too many vessels here that are piled up. We call that microvascular proliferation. That's why the imaging showed that contrast enhancement. And then when we see this on our slides, we call this a grade 4 tumor without knowing anything else. This is enough to call something grade 4. But we still do the same stains, and IDH is negative. ATRX is retained, so probably not mutated. P53 is patchy, so it's probably not mutated. And look at that proliferation. It's significantly higher than the other tumors, right? So every brown as opposed to the blue, basically percentage-wise, are the cells that are rapidly proliferating. It was 1 to 2% in low-grade tumors. Now we are up to, 15. So this is a glioblastoma, IDH wild-type, WHO grade 4. This tumor, we can get to this diagnosis in an older adult just with these stains. We don't really have to do sequencing. But if we were to do sequencing, we expect these molecular alterations. And it's very important to know what these are, mostly because sometimes we have a small biopsy that doesn't have all the features, but has all the right genetics. So we still call that glioblastoma. And, IDH is very important. Why are we not doing IDH sequencing on this case? Stain is negative, but I showed you an example. Stain is not like answered at all. We could, and we do at UCSF, sequence all tumors, but if we just look at it from an epidemiologic point of view in an older patient, finding some IDH mutation is less than 1% chance in that group. But we do sequence because that 1% is still important. And if we have the means, and we are lucky to be at UCSF, we do sequence on all cases. In addition, we sequence, and this is just like a random example, but we sometimes find other alterations, like the EGFR V8 variant, for example. And those alterations can be targeted with specific ongoing clinical trials. They may not be available as like a routine treatment, but we have clinical trials specifically targeting those variants every once in a while. And that's why this is basically the basis of personalized medicine. And finally, you may have heard about something about MGMT promoter methylation. So why do we care about MGMT? This is probably the most common genetic test. MGMT gene, so with most of the chemotherapy we give, including the Temodar, it causes multiple mistakes in the DNA. As the tumor is trying to replicate itself, it's making mistake after mistake. And our hope is that that cell with a lot of mistakes will die. But all cells, including the tumor cells, has a mechanism to correct those mistakes. So our best hope is that that mechanism is not working. So if there is an MGMT promoter methylation, that's what we specifically look for, that MGMT is not available to fix those mistakes and the cell will die. But if there is no promoter methylation, that MGMT is going to fix all the mistakes that we created with our chemotherapy, and then cells may not die as much as we want them to die. So this is a very predictive test, and we do that for all gliomas, especially predictive for glioblastomas. And you can see the response to treatment, right? If the MGMT is unmethylated, they don't respond well, but if the MGMT is methylated, they do. So this is one of the tests that really, I'm not going to say they don't necessarily give Temodar if it is unmethylated, but I think the expectation is we'll have to have something ready if the tumor doesn't respond as well. I'm actually going to stop here and see if you have any questions for me. Let me see.
I have a question. So in my mind, I think of a pathologist as someone who sits behind a microscope and the cells, you get the tissue and you look at it. Can you tell us a little bit about what happens after, like what really happens after that tissue leaves the OR?
Yeah.
Does it go through a machine or are you?
Both. So usually a portion comes directly to our lab and then we take a look immediately to see, especially if the surgeon has a question about, tell me if it's high grade, low grade, I'm going to do something different right now. So we call that frozen section. So that comes directly to us. But if not, it still comes to the lab and put into small containers, we call them cassettes, and then go through a lot of chemical processes in a machine. And someone cuts a really thin section and put it on a glass slide and then color it for us, right? For us to see. That would be those pink and purple. So H&E, we get that, and we look at them actually under a microscope. So yeah, you're right. Pathologists are usually hiding behind their microscopes, but nowadays behind our screens, because those are digitized and we can look at them on a screen now. And then next thing is basically we decide what else needs to be done.
And how long does it typically take for the whole
thing?
Yeah.
So if it is only H&E and couple immunohistochemistry, I would say maybe 4 or 5 days. Just the processing itself is overnight, and you order something that's overnight. So the moment we add things kind of in sequence, it adds up maybe 4 or 5 days. But if you keep adding more, it will be more.
Right.
Yeah. But the intro-op, for example, how the surgeon sends this piece of tissue, and we can tell them at least a preliminary diagnosis, that's about 20 minutes. Yeah.
Is there a minimum of tissue that you need in order to diagnose it?
Yes. So let me think how to say this. So if you are just going to do stains, each stain requires a section, which is like 5 microns, which is nothing. Like we can make hundreds of sections consecutively if we were to do that. But the moment we are talking about sequencing, we really want 1 centimeter by 1 centimeter by some tissue. We could do with less, but then, the lower the amount of tissue, less sensitive sometimes the tests get. So it will become, especially for a small biopsy, it becomes a triaging problem. Which test is most important? Yeah.
Does, I know you said you take into account other factors when you're trying to make your prediction. Do you ever look at gender or age and like right away you kind of have a...
Gender, not so much. Age, for sure we look at. We also have a saying in pathology, it's location, location, location. We also look at where in the brain we are at. Because if you see the name, diffuse midline glioma, it has to be in midline, for example. So we definitely look at the imaging because we want to make sure what they were expecting is what we are seeing. If there's any disconnect, it could be a random, like tumors don't really read books, they could do weird things, but we want to make sure we are not missing something. So we look for any disconnect. So radiology, location, age. Yeah.
And then if and when the tumor recurs, you do this all over again?
Yeah. Pretty much.
Because it can change.
It can change. We start with the knowledge of like what it was before. So maybe not do, if we needed to do more, maybe we don't have to do everything. But to a minimum, we look for the proliferation markers. And if it doesn't look, we also pull up the images or the slides from the prior tumor. And if it's changed significantly, we do more stains. But molecular can definitely change as tumors can also progress, but also the chemotherapy we gave them changes their DNA. So we definitely repeat the molecular unless it was done like within a couple of months, we repeat them.
Some of the research that's being done is where one of the labs, we take 10 samples from the same patient and analyze it because you can find that there's differences. So, we think of a tumor and you look at the scan, it looks like a static thing, but it got there in many different ways. So, and there's all these branches that it can take. So some may have this feature in this area of the tumor and may have a different feature in another area of the tumor, which speaks to how challenging it is to treat. Because if you sample this piece and you think, oh, I know what this tumor is, we're going to treat it with this information, but then you have another piece that's got something else, your treatment may not work as well, right? Because that's going to escape treatment. So it's very, very fascinating for that. And then, like you said, over time, what ends up emerging at the time of recurrence can be influenced by time, but also influenced by the treatment. They have radiation and the chemotherapy. That can influence the mutations and changes as well. So we're tracking 10 of those as well at the time of recurrence.
Does the immunotherapy change it too?
Yeah. So immunotherapy doesn't get into brain as well, and many of these tumors don't respond well, but when they do, immunotherapy really changes the tumor niche. Maybe not so much tumor DNA itself, but like how the tumor cells look and behave for sure.
And that's a really good point because when you're looking at this, you're looking at the tumor, but the tumor doesn't just exist as a tumor. A tumor has all kinds of cells around it that support it, and that's called a microenvironment. So, and that can change because of treatment as well. So if we're only thinking about the tumor, but there's all these other cells that are not tumor cells that are there, and how do we target that? And that's part of some of the work that Sean Kirby-Jumper is doing, looking at the neurons in the brain and how they connect to the tumor and actually excite the tumor as well. So it's really fascinating how these cells exist in the brain and the interactions that are happening in it.
Yeah, so these are neurons surrounded by all these tumor cells, and they are not just like there, they actually connect and talk to each other. So there's some kind of feedback loop going between the tumor and the normal cells in the background.
For patients that have inoperable tumors, would you be working with like a liquid biopsy or instead of a solid sample?
So we still use solid samples heavily, but liquid biopsy works. So when we say liquid, for example, rather than the tissue itself, you can get CSF, or better yet, sometimes you can just look at the markers in the blood. Because if there are enough alterations in the tumor, that DNA sort of sheds off to the fluid. So we do that. And it doesn't have as much DNA as the tissue itself, but especially if you have specific targets in mind, it works amazingly. So we do that.
So everything now is AI. How is AI influencing or affecting how your workflow or integrating it or using it in pathology?
So this H&E is the oldest pathology technique ever. It's like maybe 200 years old. Very little changed. We know our classifications changed. What AI is doing is looking at these H&Es, because we scan them, right? I told you we digitize these now. We look at them on the screen. AI algorithms can look at existing slides with their diagnoses. And if you give a new one, it can predict what that tumor is. So not as good as a pathologist yet, partly because tumors are, and especially not for brain tumors, mostly because tumors are very different from each other. But they are, actually AI is really, really good for other more common tumors, let's say prostate, for example. But what we are actively using a lot is all that 500 something genes, very few, like it's not something a human can do. Like there's not enough time to look at individual ones. So that's all AI. There are some pipelines that basically says, I compare this library with the existing library, these are abnormal. So that's AI. Or the one that I mentioned, DNA methylation. They look up the DNA methylation pattern, and I'm talking about 400,000 loci, and compare it to the existing library of hundreds and thousands of tumor with as many loci, right? So this is not something humans can do. This is all AI.
So in some regard, you've been using it for a while.
Yeah. So I think there's this topic about like, are we going to lose our jobs because AI is going to do a better job? Eventually, but probably not even my lifetime. I'm kind of mid-career. But I think we are doing AI-assisted diagnosis more and more. And I think it's definitely is the future.
And based on like these 500 genes that you test, does that help you predict kind of the tumors that eventually come back, or how they evolve and all that kind of stuff?
So most of the diagnoses, like we have a separate diagnosis. Like there is a reason why we give different names to different tumors. It's kind of embedded, right? So we know an oligodendroglioma is not going to come back as fast as an astrocytoma, and neither of them will come back as quickly as a glioblastoma, right? But even within the same tumor type, we potentially predict which one will respond to a particular treatment, temozolomide or immunotherapy, based on the molecular alterations. I mean, nothing is 100%, but it definitely gives you a better idea about which treatment to pick.
So to build a little bit on what Jen was asking about, gender or sex, even though you don't look at that, the normal genomic aspect of the patient. So what we're looking here is a tumor, right?
Yeah.
That's what's called somatic. But every patient comes with their own normal genes that make it up, but some patients have genetic predisposition to tumors too. So are you ever taking that into account? Like if you know a patient has a certain type of tumor, might you look for a certain kind of germline, it's called, because that could affect the family.
Yeah.
Like the children or the parents or so forth. So is that something you're doing?
We do, and we do very routinely. So it's either we know already because some astute clinician actually asked about family history, or sometimes we don't, but the tumor has this particular look to it that we get suspicious. And we have, again, some markers. P53 is one of the genes that could be altered in the body, like in all cells, it could be altered. And then those tumors look somewhat slightly different, so we know that. There is not an easy way of testing it other than saying you need to get your blood tested for that particular mutation. But there are other genes that normally basically fixes those small mistakes in the DNA. And if the patient's entire patients, like all cells I'm talking about, has a defect there, they cannot fix those small mistakes. We call that mismatch repair deficiency. If that happens in the tumor, there is a way of testing that. We can again do another IHC, and we can show that that protein is lost. And if that protein is lost in the tumor, there is a chance it may have been lost elsewhere in the body as well. So we also recommend please do a germline testing, because if that's the case, that patient and the family members have a tumor syndrome, like they are prone to develop more tumors because of that. So we do that. So not so much for gender other than maybe meningiomas, but for most other tumors, we look at everything else but gender.
What excites you in the foreseeable future in terms of directions these diagnostic techniques will take?
I think the kind of more specific we could be about what we are seeing, more likely there will be a specific treatment for that tumor. I think that's the goal, like this IDH mutant tumors, rather than just calling everything it's a high-grade glioma and there is one type of treatment. So I think that's what excites me. So the more we know about their molecular background, better we can target them, I think.
It's kind of interesting because as we subset tumors, you're getting smaller and smaller and smaller populations of patients to be able to study. Yeah. And so the challenge of doing neuro-oncology clinical trials is already a problem, because we're not like, it's not like breast cancer and prostate cancer, which is very, very common, very easy to sort of test new treatments as they come along. We already are challenged because the number of brain tumor patients is not very much. That's good.
Thankfully, yeah.
But on the other hand, if now we're calling 10 tumors, 10 different tumors, how do we ever test treatments for these, right? And that's a major challenge in general for us. So just something, it's exciting, but it comes with its own kind of like, how do we do this in the right way? So.
Thank you so much, Melike.
Thank you.